Hi, I'm glad to see you on Histology video lecture, and today our topic is overview of immune system histology. So let's start. Immune system is a network of cells, tissues, and organs that work together to protect the body from foreign antigens. The human body provides an ideal environment for many microbes such as viruses, bacteria, fungi, and parasites, and the immune system prevents and limits their entry and growth to maintain homeostasis. The immune system recognizes and responds to antigens. Antigens are substances, usually proteins, on the surface of cells, viruses, fungi, or bacteria. Non-living substances such as toxins, chemicals, drugs, and foreign particles can also be antigens. The immune system recognizes and destroys substances that contain antigens. Also, endogenous antigens like cancer cells can also be antigens, and our immune system is responsible for cancer antigens also. Classification of immunity. We have innate and adaptive immunity. For innate immunity, it's important that is a rapid answer. When pathogens invade tissues, rapidly reacts non-specific innate defense mechanisms like inflammation, phagocytosis, soluble molecules, and physical barriers. Examples: physical barriers like skin, mucous lining of digestive tract, respiratory passages, etc. There are several types of soluble molecules like complement and cytokines. Cytokines like interleukin, interferons, tumor necrosis factors, and others require several days specific adaptive defense mechanisms. Acquired adaptive immunity is classified into cell-mediated immunity and antibody-mediated or humoral immunity. Firstly, we will characterize innate immunity. It can be cell-mediated or humoral. Innate or non-specific immunity is a defense system with which you were born. It protects you against antigens. All antigens, non-specific. Innate immunity involves barriers that keep harmful materials from entering your body. These barriers form the first line of defense in the immune response. And we say that immune response has two stages: early non-specific stage and late specific stage. Examples of innate immunity include, for example, cough reflex, mucus production, mucus which traps bacteria and small particles, for example, in respiratory tract, in airways, skin as a barrier, lysozyme, antibacterial enzyme in tears, skin oils, and saliva, and also acidic environment of stomach and vagina. Also, examples include the body's complement system and substances called interferon and interleukin-1. Last, causes fever. Complement is cascade activated by classical pathway involving antigen-antibody complexes, or alternative pathway direct activation by invader. Initiate vascular changes, dilatation, and increased permeability. Cell lysis, opsonization. Opsonization means coating invader with complement, enhancing phagocytosis, chemotaxis, and activation. Early stage of immune response includes inflammation process. Inflammation can be chronic or acute. Vascular changes, including capillary dilatation, enhanced permeability, and increased blood flow, resulting in production of fibrin-rich inflammatory exudate. The reason of these changes is degranulation of mast cells and secretion of them histamine and serotonin. It gives rise to migration neutrophils and later macrophages. Phagocytosis, enhanced by immunological mechanisms, destroyed tissue replaced by capillary-rich granulation tissue, slowly replaced by fibroblast proliferation and collagen deposition, formation a vascular and a cellular fibroscar during chronic inflammation. Adaptive or acquired immunity refers to antigen-specific defense mechanisms that take several days to become protection and are designed to remove a specific antigen. This is the immunity one develops throughout life. There are two major branches of the adaptive response: humoral immunity and cell-mediated immunity. Humoral immunity involves the production of antibody molecules in response to an antigen and is mediated by B lymphocytes. B lymphocytes differentiate into plasma cells in peripheral immune organs, and plasma cells secrete antibodies. Cell-mediated immunity involves production of cytotoxic T lymphocytes, activated macrophages, activated natural killer cells, and cytokines in response to an antigen and is mediated by T lymphocytes. Classification of antigens: there are exogenous and endogenous antigens, are molecules that elicit an immune response in the body. Chemical nature of antigens: they can be proteins, polysaccharides, conjugates of lipids with proteins, lipoproteins, or polysaccharides, glycolipids. Antigen presentation can be present in two ways, two mechanisms: for exogenous antigens and endogenous antigens. Initial immune response to any antigens requires the antigen be recognized by a T cell. This is best exemplified by AIDS when organism loss of CD4 plus T cells. Two categories of antigens are processed and presented to T cells by different mechanisms: for exogenous and endogenous antigens. You can see that antigen presentation cell, it can be macrophage, dendritic cell, B cell. Antigen is phagocytosed by antigen presentation cell, and antigen processing occurs inside cytoplasm in lysosome, and then antigen determinant is presented in complex in connection with class II histocompatibility molecule is presented to CD4 positive T cell. For that reason, we call macrophages, dendritic cells, and B cells antigen presentation cell. And CD4 positive T cell is a cell of adaptive immune system that activates immune reaction. After activation of CD4 positive cell, T cell activated, cytokines released, cytokines released T helper 1 or T helper 2. One CD4 positive T cell releases T helper 1 cytokines. We have activation of cell-mediated immunity. When releasing T helper 2 cytokines, as a result, activation B cells and humoral adaptive immunity. Mechanism of endogenous antigen presentation. Antigens that are generated in a cell, for example, viral proteins in infected cells or cancer cells. Nucleus, viral DNA in nucleus of host cell, then it produced viral RNA in infected cell, and transcription in ribosomes, we have producing of viral peptides. Then viral peptides in complex with class 1 histocompatibility molecule is presented to cytotoxic T cell, to CD8 positive cell. Because cytotoxic T cell has a T cell receptor for class 1 histocompatibility molecule. Stages. Antigens that are generated in a cell, viral proteins in infected cell, cancer cells. Peptides displayed at cell surface, nestled in with a class 1 histocompatibility molecule. This complex is recognized by CD8 positive T cells. CD8 positive T cells are cytotoxic. We call them also T killers and have machinery to destroy infected cell. T cell activated, proliferation of cytotoxic cells, and cytotoxic cells kills infected cells with this virus. Specific role of B lymphocyte. Summary, I can say that B lymphocytes are cells of humoral adaptive immunity. They proliferate, they differentiate into plasma cells. Plasma cells secrete antibodies. This is humoral type of adaptive immunity. But B lymphocytes can be also antigen presentation cell. Process antigen by myristocompatibility complex 2 pathway, but B cells engulf antigen by receptor-mediated endocytosis. They have B cell receptor, this one. Antigen is bind to B cell receptor. Then antigen processing in lysosome. And antigen determinant is presented in complex with class 2 histocompatibility molecule to T cell receptors, helper T cell, CD4 positive cell. B cell receptors are surface antibodies encoded in plasma membrane. Affinity of B cell receptor for an antigen epitope is so high that the B cell can internalize the antigen at concentrations thousands of times smaller than needed for a macrophage. CD4 plus T cell recognizes displayed antigen and is stimulated to release cytokines. These stimulate B cells to grow into a clone of cells, plasma cells. These plasma cells synthesize B cell receptors with identical binding site for the antigen epitope, but the transmembrane tail. The antibodies are secreted as a result of this mechanism. When B lymphocytes are antigen-presented cells, they activate only humoral adaptive immunity response. You can see simple schematic diagram of immune response. Ingested pathogen in antigen-presented cell, you can see helper T cell recognizes this antigen. Helper T cell produces cytokines and activates or humoral immunity or cell-mediated immunity. Clonal expansion. Look, please, what happened after activation of cytotoxic T cell? Activated cytotoxic T cell. This is effector cell of cell-mediated immunity. The activated T cell increases in size and divides by mitosis. And a clone of competent T cell is produced. They proliferate via mitosis, and T cell differentiate, becoming various types of T cells. And we have helper T cells, cytotoxic T cells, and memory cells. This is mechanism of cell-mediated adaptive immune response via clonal proliferation. B cells, humoral immunity mechanism, proliferation via soluble and due to soluble antigens. Primary response, initial encounter with antigens, proliferation to form a clone, subsequent challenge by same antigens, and then when it occurs, secondary response can be years later, for example, clone of cells identical to ancestral cells. And plasma cells are, we have as a result clone of plasma cells, and plasma cells produce antibody against this antigen. This is specific humoral immunity response. But now immune cells, immunocompetent cells. What cells of our body can take part in immune response? Firstly, blood form elements. You can see blood smear and granulocytes, neutrophil, also eosinophil and basophils. Do you remember that granulocytes, that leukocytes together, they act only in loose connective tissue, but in bloodstream only transport, only migrate. After some time in bloodstream, they migrate to loose connective tissue and do their functions in loose connective tissue. Neutrophils, eosinophils are reason to inflammation, give rise to inflammation, and basophils differentiate in loose connective tissue to mast cells. And mast cells also take part in inflammation, non-specific stage of immune response. Next, monocyte. Monocyte also is a granular leukocyte. Monocytes migrate from blood to loose connective tissue and in different organs and differentiate into macrophages. Macrophages take part in non-specific stage of immune response. For example, they are phagocytic cells. And they take part also in a specific stage of immune response. They are antigen presentation cells. Types of macrophages. It can be dendritic cell. It can be interdigitation cell, Langerhans cell, and in different organs, for example, in liver, Kupffer cells, in lung, alveolar macrophages, in nerve tissue, microglial cells. All these cells are macrophages and they take part in immune response. Okay, and lymphocytes. Remember that lymphocytes are cells only specific immune response, only adaptive immune response. And we have some types of lymphocytes, T cells and B cells. B cells are differentiated in red bone marrow and then migrate to B zones of B-dependent zones of peripheral immune organs, but T cells must firstly have differentiation and selection in thymus, and only after thymus they migrate to peripheral immune organs like spleen, lymph nodes, tonsils, and they are settled down in T-dependent zones of these organs. B cells and T cells provide adaptive immune response. This diagram demonstrates you non-specific immune response via action of mast cells. We have injury of tissue. Next, tissue damage. Microorganism acts and as a result we have tissue damage. Microorganism, what pathway? It can be mast cell activation, degranulation. And after degranulation, mast cell release histamine. Histamine acts to blood vessels wall. And as a result, vascular permeability increased and decreased vascular inner diameter, and then prostaglandins and leukotrienes are released by endothelial cells, and then we have inflammation process in tissue, inflammatory process. Next, polymorphonuclear cells. It means neutrophils, eosinophils, and basophils. Mostly eosinophils and neutrophils. They provide phagocytosis, but we called them microphagocyte cells. And then we have fluid exudate and oedema in loose connective tissue in this place. Comparing B cells and T cells, we can note next important points. B cell, a type of white blood cell, and specifically a type of lymphocyte. Many B cells mature into plasma cells and produce antibodies necessary to fight off infections, while other B cells mature into memory cells. All of the plasma cells descended from a single B cell produce the same antibody, which is directed against the antigen that stimulated it to mature. The same principle holds with memory B cells. Thus, all of the plasma cells and memory cells remember the stimulus that led to their formation. The maturation of B cells takes place in birds in an organ called the bursa of Fabricus. And that's why we called B lymphocytes, B cells. B cells in mammals mature largely in the red bone marrow. The B cell or B lymphocyte is thus an immunologically important cell. It is not thymus dependent, has a short lifespan, and is responsible for the production of immunoglobulins, or we can say antibodies. It expresses immunoglobulins on its surface, and these immunoglobulins can act like B cell receptor. Compare with T cell. T cell is a type of white blood cell also that is of key importance to the immune system and is the core of adaptive immunity, the system that tailors the body's immune response to specific pathogen. T cells are like soldiers who search out and destroy the targeted invaders. They have TCR on the surface, T cell receptor. This receptor is formed in thymus. Immature T cells, stomped T stem cells, migrate to the thymus gland in the mediastinum, where they mature and differentiate into various types of mature T cells, and become active in the immune system in response to a hormone called thymosin and as other factors. T cells that are potentially activated against the body's own tissues are normally killed or changed down-regulated during this maturation process. There are several different types of mature T cells. Not all of their functions are known. T cells can produce substances called cytokines, such as the interleukins, which further stimulate the immune response. For example, T helper cells, CD4 positive cells, cytotoxic T cells, or T killer cells destroy virus-infected cells and tumor cells and are also implicated in transplant rejection. They provide anti-transplantation immunity. These cells are also known as CD8 plus T cells because they have CD8 glycoprotein as their surfaces. In this slide, you can compare T lymphocytes functions, T helper cells functions, and T cytotoxic cells function. I'm sorry for this mistake. It must be cytotoxic, but not epsilon. Yes, I'm sorry. CD4 positive T cells. When a CD4 helper T cells binds molecular histocompatibility complex 2 and antigen complex on an antigen presenting cell, both the antigen presenting cell and the T cell release cytokines. Next, in response to cytokines, the T cell clones itself. The cloned T cell produces different cytokines that activate B cells and CD8 positive cells. And T cytotoxic cells. When the cytotoxic T cell interacts with the myristocompatibility complex 1 epitope complex on an infected cell and produce granzymes and perforins. Granzymes and perforins are proteins. The perforins form pores in the plasma membrane. Granzymes enter the cell and break down proteins, releasing the cell. Infected cell is destroyed as a result. On this slide, you can see scanning electron microscopical view of B cell and T cell function. B cell differentiating into plasma cell. Plasma cell products immunoglobulin, and immunoglobulin provide humoral immune response. Antibody connects to antigen and form complex antigen-antibody, and as a result, macrophages phagocyte this complex and eliminate it. And T cell, T lymphocyte, has TCR receptor and provide cellular immunity. It about T cytotoxic cell. And T helper cell, you know that it activates or humoral immunity response or cell-mediated immune response. You can compare mechanism of humoral immunity with cellular immunity. Humoral immunity is develops into extracellular microbe, for example, bacteria. You can see B cells. B cells have receptor B cell. The receptor, this is immunoglobulin. To immunoglobulin, to this receptor bind antigens, then this next we have neutralization. This is the first way, and lysis by help of complement. And others way, B cells phagocyte antigen, and then look please, macrophages can phagocyte antigen and destruction of phagocytosis microbes this way. And cellular immunity develops in response to intracellular microbe, for example, viruses, antigen presentation cell, for example, macrophage, and it presents information about antigen to helper T cell. Helper T cell have TCR, and then helper T cell secretes cytokines. Proliferation and activation of effector cell, macrophages, cytotoxic cell, and as a result, also killing of infected cell or destruction of phagocytosis microbes. The development of T and B cells. The central lymphoid organs where lymphocytes develop from common lymphoid progenitor cells are labeled in yellow in hematopoietic tissue of red bone marrow. This one you can see. And thymus. This is central organs of development of T and B cells. The common lymphoid progenitor cells develop from multipotent hematopoietic stem cells in the bone marrow. Some of the common lymphoid progenitor cells develop locally in the bone marrow into immature B cells, while others migrate to the thymus T stem cells via the bloodstream, where they develop into thymocytes, developing T cells. T cells and B cells are activated by foreign antigens, mainly in peripheral lymphoid organs such as lymph nodes and spleen and tonsils, and lymphoid tissue associated with mucus. And only in peripheral immune organs we have T cell mediated immune response and antibody response. This is adaptive immune response. So, lymphoid organs are classified into primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs are where lymphocytes are formed and mature. They provide an environment for stem cells to divide and mature into B and T cell. Both T cell and B cell are born in the bone marrow. However, whereas B cells also mature in the bone marrow, T cells have to migrate to the thymus, which is where they mature in the thymus. Multiplication is not dependent on the presence of antigen. So, we can say that in primary lymphoid organs, central lymphoid organs, antigen-independent differentiation of T and B cells occurs. Compare with secondary lymphoid organs or peripheral lymphoid organs. are arranged as a series of filters monitoring the contents of the extracellular fluids, for example, lymph, tissue fluid, and blood. The lymphoid tissue filter in each of these fluids is arranged in different ways. Secondary lymphoid tissue are also where lymphocytes are activated. Secondary lymphoid organs include lymph nodes, tonsils, spleen, Peyer's patches, and mucosa-associated lymphoid tissue. And the most important difference is that multiplication is dependent on the presence of antigens. And we can say that antigen-dependent differentiation occurs in secondary peripheral lymphoid organs. Lymphoid tissue common overview. A lymphoid tissue is connective tissue characterized by a rich supply of lymphocytes. Free within the regular connective tissue or is surrounded by capsules forming the lymphoid organs. Thymus, spleen, lymph node. Is dark blue in hematoxylin and tyrosine stain. Stroma of lymphoid tissue is reticular connective tissue. Do you remember this is a type of connective tissue with special functions? And reticular connective tissue consists of rich network of reticular fibers, type 3 collagen, and reticular cells. We have exception. In thymus, stroma consists of reticuloepithelial cells. They have origin from ectoderm, but reticular connective tissue is derived from mesoderm. Free lymphoid tissue or encapsulated. These are mucosa-associated lymphoid tissue, tonsils, Peyer's patches in the small intestine. Here you can see, for example, lymph node tissue, light optical view of lymph node tissue, and we can see subcapsular sinus. This is subcapsular sinus, and we can see macrophages in subcapsular sinus. Macrophages are antigen-presenting cells. They uptake antigens from lymph and present information about antigens to T helper cells in lymph node. Primary lymphoid organs. And we will characterize firstly bone marrow. In Latin, medulla ossum rubra. You can compare localization of red bone marrow in childhood and adulthood. In all bones, red bone marrow present in children, but in adult human body, you can see only these bones, not legs, not arms. Histological structure of red bone marrow. It consists of stroma and parenchyma. Stroma of red bone marrow is reticular connective tissue with blood vessels and type of blood capillaries is sinusoid type. With fat cells, with reticular cells and reticular fibers. And parenchyma. Parenchyma consists of lymphoid and myeloid tissue. Light optical view of bone marrow. We can see bone trabecule with red bone marrow, and you can recognize B and T cell precursors, stromal cell, reticular cell. This is megakaryocyte, precursor for platelets, and fat cells. Also light optical views of red bone marrow. stained by hematoxylin and eosin. Stroma, blood capillaries, fat cells, adipose cells, and lymphoid and myeloid tissue. Look on the left side, you can see bone trabecule, granulocyte islets, megakaryocyte, precursor for platelets, erythroid island. Formation of lymphocytes in red bone marrow. We have blood stem cells. From blood stem cells, this is pluripotent cells, polypotent, are derived two types of cells, myeloid stem cells and lymphoid stem cells. Next, myeloid stem cells is differentiated into next ways: red blood cells, platelets, granulocytes, and monocytes. Lymphoid stem cell is derived to lymphoblasts and lymphoblasts into B lymphocytes, T lymphocytes, and nature killer cells. Lymphocytes, monocytes, and granulocytes we called white blood cells. Next, thymus. This is bilateral organ. It is located in mediastinum and provide T-cell maturation. Thymus has unique stroma structure. Epithelium of thymus is derived from the third pharyngeal pouches invaded by lymphocytes. You can see in embryo. And compare please topography and localization of thymus and size of thymus in newborn, close to heart, very big size, and in adult. One more time, development from the caudal part of the third pharyngeal pouch. This one, thymus. This one. So, thymus stroma has ectoderm origin. Thymus histology. Thymus has stroma and parenchyma. Stroma consists of connective tissue capsule, trabeculae, and trabeculae divide organ into lobules. Each lobule contains cortex, peripheral cortex, and central medulla. Cortex stains more darkly. Centrally located medulla is more lightly. What happened in thymus with T stem cells? T cells precursors rearranges in its T cell receptor gens in the thymus. First process is arrangement of T cell receptor. Immature T cells that recognize self-Mayer histocompatibility complex receive signals for survival. Those that interact strongly with self-antigen are removed from the repertoire. So, T cells that act with self-MHC, this is Mayer histocompatibility complex type 1, they must be phagocyted by macrophages and eliminate. Mature T cells encounter foreign antigens in the peripheral lymphoid organs and are activated. Activated T cells proliferate and migrate into peripheral sites to eliminate infections. So we can say that in thymus, selection, first process is formation of TCR, T cell receptor, and then process of selection. Selection has two stages. First, positive, second, negative. Positive selection. T cells with TCR must be available to react with foreign cells, and reticuloepithelial cells demonstrate them some histocompatibility complex, and then T cells will react. If T cells react, this is correct cells. False cells don't react with reticuloepithelial cells, and false cells must be eliminated. This is first stage of selection, positive. Negative selection. Macrophages demonstrates to T cells information about self-antigens, so histocompatibility complex type 1. If T cells react with macrophages, then these cells must be eliminated. Why? Because they will react in future in the human body with self-antigen, and it will be reason for autoimmune diseases. So false T cells that react with self-antigens must be eliminated. Other cells stay. So as a result, from thymus, only 5% of activated T cell migrate into peripheral immune organs. Only 5%. This is a light optical view of thymus. You can see cortex, peripheral cortex, and medulla. Capsule, cortical epithelial, reticuloepithelial cell, this one. Reticular epithelial cell form network, and in lots of this network, T lymphocyte mature and proliferate and differentiate. Mature and differentiate. You can see cortical reticuloepithelial cells, and medullary reticuloepithelial cells. Also in thymus present dendritic cells and macrophages. Reticular epithelial cells are nurse cells. They provide barrier function, trophic function, and they help in maturation of T cells. You can see blood supply in thymus, in cortex, and in medulla. Cytoreticulum, achromatical epithelial nuclei, desmosomes, blood-thymus barrier in the cortex is located. Hassal's corpusculus contain keratin, calcification, and these are old reticuloepithelial cells. And note that thymus has only efferent lymphatics. Thymus lymphatics, medulla, intertrabecular, intrabecular, in cortex. And this is light optical view of thymus. Note capsule, trabecule, cortex, medulla. In medulla, you can see Hassal's corpuscle. And with age, in adulthood, in older people, there is thymus adiposus, and tissue of thymus is changed into adipose tissue. You can see cortex is adipose tissue in cortex, and in medulla there are more cells corpuscles. Next part, secondary lymphoid organs. Firstly, we will characterize lymph node. Bean-shaped encapsulate structures distributed through the body along the course of lymphatic vessels. In-life filters that are important in the body's defense against microorganisms and the spread of tumor cells. Lymph tissue fluid is filtered by at least one node before returning to the circulation. Structure of lymph node. Lymph node has stroma and parenchyma. Stroma includes capsule, dense irregular connective tissue capsule, trabecules, and also reticular connective tissue with reticular cells and reticular fibers. Convex surface. Entrance for lymph, efferent lymph vessels. This one. Efferent lymph vessels. Concave depression forms hilum. Efferent lymph vessels, arteries, nerve veins. Parts of the lymph node. Peripheral cortex, this one. Middle paracortex, this one. And centrally located medulla. Unit of the lymphatic tissue. In cortex of lymph node, lymphoid nodules are located. And we can recognize primary lymphoid nodules and secondary lymphoid nodules. Primary lymphoid nodules are without germinal center. Secondary lymphoid nodules are with germinal center. You can see this histological section, secondary lymphoid nodules with light germinal center. Found lymphoid nodules found in secondary lymphoid organs or as solitary lymph follicles, usually under epithelia, in tonsils and mucosa, has spherical structures, contains mainly B cells. Remember that lymphoid nodules are B-dependent zone. Site of B lymphocytes activation by antigen-bearing macrophage or follicular dendritic cells. Have germinal center, centrum germinativum, site of B lymphocyte proliferation, pale staining, and in the center, this one, germinal center, and marginal zone, more darkly. Location. They can be free. They are located in lymph nodes, in spleen, in tonsils. But note please, not in thymus. Not in thymus. In the cortex are located reticular cells, macrophages, antigen presentation cells, dendritic cells, B lymphocytes. They have myristocompatibility complex second type on their surface. Lymphocytes, of course, and cortex is arranged by lymphoid nodules, and between them interfollicular areas, subcapsular sinuses, this one, subcapsular sinuses, and cortical sinuses or intertrabecular sinuses. Lymph migrate from efferent lymph vessels to subcapsular sinuses and next to intertrabecular sinuses. And antigen presentation cells uptake antigens from lymph and present information about them to lymphocytes in cortex in lymphoid nodules. Cortex is B-dependent zone. Paracortex is located deeply. No nodules, no B cells, but T cells mainly in paracortex. Also present interdigitation dendritic cells, T cells. In venous, lymphocytes enter across the vessel's wall. This is venous with high endothelium. And medulla, centrally located light medulla. In the medulla, medullary cords and medullary sinuses are located. Medullary cords consist of B cells, plasma cells, macrophages. Medullary sinuses are located between medullary cords. So, cortex and medulla are B-dependent zones. Paracortex is T-dependent zone in lymph node. Schematic diagram of lymph node. Efferent lymphatic vessels carry lymph to cortex. Firstly, subcapsular sinuses, this one. Next, intertrabecular sinuses. Next, medullary sinuses. And then efferent lymphatic vessel carry lymph further. Cortex, mainly B cells. Paracortex, mainly T cells, without lymphoid nodules. Medulla with medullary cords and medullary sinuses. Lymph flow. Efferent lymphatic vessels, subcapsular sinuses, cortical sinuses, intermediate or trabecular sinuses, medullary sinuses, and terminal sinuses. During the passage, the lymph is filtered and modified by the immune cells. Is cleared, yes? We have cleaning of lymph in lymph node. Finally, lymph is collected by the efferent lymphatics. One more time, on this diagram demonstrates you lymph circulation through the lymph node from efferent lymph vessels through all sinuses to efferent lymphatic vessels. Next, spleen. Spleen is the largest single accumulation of lymphoid tissue, involved in filtration of blood, defense against blood-borne antigens, destruction site of erythrocytes and platelets, production site of antibodies and activated lymphocytes. Any inert particles in blood are actively phagocytosed by spleen macrophages. So, we see that in our body, in human body, present three lines of barriers. First, mucosa, outermost barrier, and non-specific immune response, skin, mucosa. And next barrier is lymph. When antigens enter lymph, their lymph nodes protect us from antigens in lymph. And then when antigens enter blood, spleen protects against antigens in blood. Spleen has not only immune functions. You can see that it's place of destruction of erythrocytes, platelets, and its depot of blood also not only immune function. Spleen histological structure. It also has stroma and parenchyma. Stroma consists of capsule, dense connective tissue, but note it also has smooth muscle cells. From capsule deeply to organ go trabeculae. Trabeculae also consists of loose connective tissue and from smooth myocytes. Trabecule subdivide parenchyma and parenchyma we called splenic pulp. Splenic pulp is divided into white pulp and red pulp. Also hilum, present in hilum, and through the hilum in spleen enter arteries, veins, and lymphatics. Splenic pulp is parenchyma of spleen. Stroma of splenic pulp is reticular tissue. It consists of reticular cells, lymphocytes, blood cells, macrophages, antigen-presenting cells. Two types, white pulp and red pulp. White pulp, this one. This is lymphoid nodules and periarterial lymphoid sheets. Has purple staining by hematoxylin and thiosin. Red pulp is blood-filtered sinusoids, and splenic cords, and has reddish staining. There is light optical view of spleen, and you can see white pulp, red pulp. White pulp consists of lymphoid nodule, B-dependent zone, and periarterial lymphoid shed, T-dependent zone. Lymphoid nodule, B-dependent zone, and periarterial lymphoid shed. This is T-dependent zone in spleen. Look please, this arteriole we call central arteriole of lymphoid nodule, but in the histological section it is located peripherally in lymphoid nodule. And lymphocytes surrounded this arteriole we call, there are T lymphocytes. This is T-dependent zone. It's interesting that circulation in the spleen has two types, open type and closed type. Splenic artery, first, splenic artery, then blood stream in trabecular arteries, then in central arterioles surrounded by periarterial lymphoid sheet, this one. Periarterial lymphoid sheet receive large number of lymphocytes, B cells, and may form lymphoid nodules. Is eccentric but still called central arteriole. A smaller branch supplies the lymphoid nodule. Marginal zone, surrounding nodules, blood sinuses, lymphoid tissue, lymphocyte, and macrophages. In the red pulp, there are panicular arterioles, splenic cords. This is network of reticular cells and fibers, lymphocytes, macrophages, plasma cells, erythrocytes, platelets, granulocytes, and venous sinusoids lined by stave cells, oriented in parallel with sinusoids blood flow, sparsely grouped in reticular fibers set in transverse direction, like hoops surrounding the staves of a wooden keg. This one. White pulp. Central arteriole, this one. Periarterial lymphoid sheath, T-dependent zone, this one. Splenic nodules or Malpighian nodules, B-dependent zone. Lymphoid nodules also have germinative center, marginal zone, with loose lymphoid tissue, B cells, marginal sinuses, and macrophages. White pulp of spleen. You can see blood circulation in white pulp. Trabecular artery, next central artery of lymphoid nodule, periarterial lymphoid sheath, and then lymphoid nodule. We have immune and adaptive response, proliferation of T cells, and proliferation of plasma, B cells and plasma cells, and producing antigens in lymphoid nodules of white pulp. And red pulp. Red pulp consists of splenic cords and splenic sinuses. Blood flow in the red pulp. Two types, closed circulation and open circulation. Closed circulation, peninsular arterioles or capillaries branching from them connects directly to the sinusoids. Open circulation, other peninsular arterioles are open-ended, dumping blood into the stroma of the splenic cords. And this slide demonstrates immunohistochemistry recognition of T and B dependent regions in human spleen. On the right side, you can see CD20 positive B cells, activated B cells, and you can see that B dependent region is white pulp, is lymphoid nodule. This one. And CD3 positive T cells localized in periarterial lymphoid sheath in spleen, this one. So, T and B dependent areas in different lymphoid organs. In lymph node, cortex with lymphoid nodules, and medulla are B-dependent areas. Paracortex is D-dependent area. In spleen, white pulp, lymphoid nodules of white pulp are B-dependent zone. Periarterial lymphoid sheath is T-dependent zone. In tonsils, lymphoid nodules of tonsil are B-dependent zone, and diffuse lymphoid tissue surrounding lymphoid nodules is T-dependent zone. And a couple information about mucosa-associated lymphoid tissue. Free lymphocytes and follicles in various sites in the body where antigens are present. For example, gut, lungs, salivary glands, airways, and urinary tract. No capsule. In some places, aggregates form. For example, in tonsils, Peyer's patch, appendix. And note that 70% of the immune cells are localized in the mucosa-associated lymphoid tissue. On this histological section, you can see appendix, mucosa of appendix, and lymphoid tissue via lymphoid nodule in submucosal layer. A short information about tonsils. They are localized in the mucosa and provide local immunological protection. While there's lymphatic ring, we called six tonsils, pharynx, palate, auditory tube, root of the tongue. Epithelial covering depends on the location and lymphoreticular tissue, no sinuses. General structure of the tonsil. You can see mucosa is stratified squamous non-keratinized epithelium, invagination of mucosa. We called it crypt, and lymphoid tissue surrounded this crypt under mucosa, and we can see lymphoid infiltration with macrophages and lymphoid nodules and diffuse lymphoid tissue. Tonsilla palatina is localized in oral cavity. Epithelium stratified squamous non-keratinized. Crypts are deep. Lymphoid infiltration in the epithelium. Hemicapsule, and present also venous with high endothelium. Tonsil palatina. You can see inflammatory process in tonsil palatina, and it calls tonsillitis. After injection with antigens, with bacteria, for example, streptococcus. Light optical view of tonsil palatina. Crypt, mucosa of crypt, lymphoid tissue of tonsil, secondary lymphoid nodules, primary lymphoid nodules, diffuse lymphoid tissue. Look please that this section is stained by trichrome, by Masson, and loose connective tissue with collagen fibers is stained in blue color, this one, and this is salivary glands, secretory units of salivary glands, mucocytes you can see. Lymphoid infiltration in the epithelium of the crypt. Stratified squamous non-keratinized epithelium can be infiltrated by lymphocytes and macrophages. And this slide demonstrates high endothelial venule, its place for migration of T and B lymphocytes. Tonsilla lingualis. You can see localization of tonsilla lingualis, this one, and you can see histological section of tonsilla lingualis. Also, mucosa, crypt, lymphoid nodules, diffuse lymphoid tissue. No capsule, and crypts are not deep. Tonsilla pharyngea, impaired, difference in covering pseudostratified ciliated epithelium with goblet cells, lymphoid infiltration in the epithelium also. Crypts are deep, folliculus and interfollicular area, and lymphoid nodules or follicles are B-dependent zone, and interfollicular area are T-dependent zones. Have seen capsule and septa and seromucous acini in proximity. Peyer's patches are localized in distal part of small intestine. This is aggregates of lymphoid nodules in submucosal layer. You can see. So it's all for today. I hope that this lecture will be useful for you. I'll see you soon for studying histology. See you. Bye.